Howdy, everyone. I'm Lillian Hislop. I'm a postdoctoral fellow with the USDA in Corvallis at the Blueberry breeding program there. This talk was supposed to be tools and techniques for GWAS and genomic prediction in blueberries and breeding insight. I wrote my abstract very optimistically. And I just got my genetics fully processed last week, so I haven't actually been able to do any GWAS or genomic prediction. So this is a results so far, and then with the tools I've learned at the Tools for Polyploids meeting, hopefully we can actually apply it. So thanks for bearing with me. Today I'm going to talk about breeding insight, the project, how it's outlined, our methods, and what we've learned so far. So this project is in collaboration with Breeding Insight. Breeding Insight is a USDA initiative that is aiming to bridge the technology gap where crops and animal models like cows and corns have all these resources and these amazing tools for genomic prediction and all these cool technologies that more underserved crops don't necessarily have access to. So Breeding Insight is trying to bridge that technology gap and get blueberries kind of up to par. Breeding Insight also works with other species like cranberries, cucumbers, trout, honeybee. So I think that's neat. And it's a really great team, very collaborative. And the ways that they're helping us are kind of in two prongs. They are helping us with our genetics. BI and the Corvallis breeding program developed a marker platform through diversity array technologies with about 3,000 markers based off of SNPs and small indels. We took the genetic material back in 2021 and sent it off, and now it's gotten back and it's been all cleaned and processed. So that's really neat. They also are helping us with phenotyping tools. This summer, I was helping them trial Fieldbook, which is an open source Android app developed by the Poland lab at Kansas State University. And BI is working with them to make it a little bit more robust and work for a wider array of phenotypes and plants and things like that. Fieldbook is really convenient. It is adaptive. You can plug in your field layout and then it works really intuitively where you can walk through the field and take the phenotypes as you go. It also allows samples to be connected to QR codes, so you can just scan them, which is great when all the samples have been thrown in the freezer and then taken out at completely random orders. And it changes dynamically based off what kind of phenotype you're taking. So if you're taking a date, it has a section to put dates in and it can open the calendar. If you're doing a binary tree, it just gives you a yes or no. So it's really nice to eliminate typos. It takes who is taking the data so you can correspond it to individual technicians and factor that in. It records when all the data was taken. Overall, really happy with it. We also trialed dictation software that allows you to walk through the field and talk to the computer and take your phenotypes orally if you're working alone, which is really nice. And this all connects to BreedBase, which is a database to collect all your phenotype data in one place. So you're not working with 3 million spreadsheets. So that was all really nice. So the project that I'm working on, we took these fields that were planted in 2016 and 2017 as our breeding fields. We were going to be making selections out of these initial seedlings, but they were kind of underutilized due to the passing of Chad Finn. And so we're like, okay, what can we do to repurpose these? So we have 5,000 offspring from 85 different parental crosses, about 50 to 100 siblings per family. And as I said, they're planted in 2016 and 2017. So these are pretty mature plants. And the idea is we have all this mature plant data. Can we use this phenotypes and do genomic prediction on it to predict whether seedlings are going to be good or not and not have to bother growing them out for 7 years? So from these 5,000 offsprings, we collected plant architecture data like height and ripening time and bloom time. We collected all this data as well in 2020 and 2021. And then this summer, I was able to collect 1,400 samples with the help of my crew, which was a really nice amount of samples that I couldn't do on my own. And for these samples, we looked at things like fruit quality, things like that. So overall, we collected 16 phenotypes. Some of them were more subjective, like flavor and texture and appearance. I'm not personally trained in blueberry taste testing specifically, but I have a lot of opinions, and I do a little bit of corn, so I tried. And things like, does this seem to be visually exhibiting shock virus? And we stored samples that we harvested for 6 weeks in cold storage and looked at their decay after 6 weeks. We also had our fruit architecture that I mentioned, or our plant architecture like height, and then the 5%, 50%, and 95% bloom ripening thresholds and ripening thresholds. And then we have our more quantitative things like Brix, pH, and acidity that we took from frozen berries, so we could do them at our leisure in the fall. We took fruit diameter, mass, firmness immediately after harvest, and then firmness after 6 weeks in like storage in a fridge. So like I said, we used Fieldbook as one of our phenotyping aids. I got a little video of it working. It's pretty swifty. And then we used Otago handheld meters for taking our Brix and pH measurements and acidity measurements. Very user-friendly, just able to just take juice and apply it to it. We were really happy with the accuracy of that. And it's really nice to have crew. Oh, it's great. And then this is Freebase and kind of what it looks like. You have your different fields all accumulated in one place, and you can look at other people's phenotypes from other trials as well, which is really cool and collaborative. So our genotyping was done through dart tag with our known SNPs and small indel markers. The marker platform that we developed had about 3,000 probes. And after we got it back and accumulated it into one place, to do the processing that I'm about to outline, we separated them into kind of 13 clusters based on parentage so that we could do it computationally. If we tried to do all this processing on all of them all at once, all 5,000 samples, it was going to be too computationally intensive. So how we processed the genotypes is we filtered for missing data on both the sample and the marker level. We concatenated all the reports into one big report. We extracted all the SNPs from those concatenated reports. Then from the big concatenated report, we also filtered by missing data for the SNP and marker level on a kind of single SNP basis. We looked at the hint and he function in Polyrad, and then we did our genotype calling using Polyrad to get our genotypes and dosages and stuff like that. And then we eliminated anything that had poor signal sample. And when we were looking at the ratio between H individual and H expected, we could find some of these outliers. You can see them pretty clearly, like here. And that was one of the ways that we did our quality control and filtering. And we did this on each of the clusters. So this is what the dosages ended up looking like. The number of clusters, we had about 9,000 SNPs before filtering and about 8,000 SNPs after filtering within each of those. And you can see here the different clusters. So this was all the families that had legacy as one of the parent in the crosses. And each of these colors is a different family set. So in the raw data, you can see they're clustering pretty well. And then we filtered by hint and he, and then we estimated population structure. And you can see how the clusters kind of become clearer and there's less outliers. So we're pretty happy with the amount of quality control and filtering that we did. Here's another example of that in the ways that the clusters kind of clarify. So now that we have this nice genetics data, whoop, boop, boop, for all the samples, all 5,000 samples, I'm excited to now do the genetic analysis. So what we've learned so far phenotypically, well, excuse me, we're going to be doing our analysis with GWAS poly and stage-wise. And since I received them last week, I'm excited to process. So what we've learned so far phenotypically is these are the fruit quality traits from 2022. Overall, seems really normally distributed and exactly where we expect them to be. We're happy with the kind and quality of data that we got from our simple handheld meters. Here is our bloom and ripening thresholds over the last 3 years. We're seeing the trends that we expected to see. Last year in 2021, there was a big heat dome, and this year there was a really late spring. So we were hoping to see kind of a delay this year compared to last year, and we did see those. The blooms took a little bit longer to come into being, and the ripening, same thing with the ripening thresholds. We saw some interesting correlations. Turns out this is firmness immediately after harvest versus firmness 6 weeks after harvest. They correlate with each other, which is, what you'd hope. If it starts as a firm berry, it's probably going to end up being a firm berry 6 weeks later. We found that the amount of sugar seems to negatively correlate with how heavy the berries are, so how large they are. So the bigger they are, the less sugar is in the samples. And here is the variation in pH across the different families. So each of these box plots is a family, and they're colored by similar parentage. So you can see variation among the different things that are related, but also clustering in things that are similar. So like this has a shared parent, and you can see the trend versus this one, for example. So the genetics look like we expected to. We're excited about the filtering and the quality of them. We have a wide diversity of fruit quality traits and architecture traits that we're excited to analyze. And I'm excited to use the tools that I've learned here to look at what we predict. So lots of people go into this project, and I'm very grateful for all of them. And I will take any questions if you got any.